Oh, so this is recording.
Thank
Okay.
you. Yeah.
So we believe that actually Schwann cells have two distinct neuroprotective roles, neuroprotective functions, and maybe later you will better understand this, but I want to briefly to touch upon this as an introduction. So Jason Mirsky and other people, they think about the C-June orchestrated role to promote axonal regeneration, growing of new axons, in the distal stump, if the nerve completely degenerates, this facilitates growth of new axons. But I think a little bit differently. In many diseases, axons are only mildly injured, I would say, subacutely mildly injured. They don't completely degenerate, so you don't need to regrow new axons. You just need to help them to recuperate from small micro lesions. So, and I think my pathway, the pathway that we identified, helps axons to heal. So you don't a priori, preemptively, so you don't need really to regenerate new axons. So I think these are two fundamental different neuroprotective functions. Okay, anyway, but so thank you very much for your nice introduction, and to all organizers for inviting me, so giving me this opportunity to talk today about this axon glia metabolic crosstalk, and which role this plays for the regulation of axon stability. So my name is Bogdan Beirowski, and I know that you mentioned I work in the Hunter James Kelly Research Institute. So this was the name until 3 months ago. We changed now the name, so it's called Institute for Myelin and Glia Exploration. So how do I change to the next slide? Here. So before I jump in, just let me give you a brief outline of the seminar. So first, you will see why is it important to study axons and their integrity. Then you will learn why is it important to incorporate the biology of glial cells into the study of axon integrity. So this is really not a question that is kind of intuitive, because so far, the biology of glial cells in this context was very strongly overlooked. So most people that study like, axon degeneration mechanism, they never think about glia. So this is what I mentioned before. So it's very intriguing, very interesting to think about this. And then I will speak about our most recent story, so published in Nature Neuroscience. Schwann cells sense axon injury and adjust their glycolytic activity to support axons via axoglial metabolic coupling. So this is the bottom line of the study. And then finally, we will learn how can this knowledge from this discovery be applied to confer neuroprotection in models of acute, subacute, and also chronic axon degeneration. So, as a brief introduction, I wanted to show you some pictures from like classical neuroscience textbooks or websites that illustrate neurons. So neurons, as you know, are are highly compartmentalized cells with, this dendritic tree, the cell body compartment, very different than other cells. And in between here is always the axon. But if we look at such pictures, we have to ask ourselves, do such pictures really illustrate how axons are in reality? They're certainly not, so for two major reasons. First, axons do not exist in isolation, as you know, but they're intimately associated with, I like to call them enwrapping glia, which they form symbiotic relationship. So this enwrapping glia are Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. So I call them axon flanking glia. So these cells are best known, or are traditionally known, for forming compact myelin sheaths around axons to facilitate impulse conduction, as you see here. But a fact that is often neglected is that many axons in the PNS and also in the CNS are actually not surrounded by myelin. So here you see a cross-section electron micrograph of the vagus nerve. The majority of the axons are not myelinated. And this indicates another and more ancestral function of these enwrapping glia, which is the trophic support of axons and the subject of my seminar today. So another key realization that is not clear from the simplified picture I showed before, that axons are extremely long and thin structures with a peculiar cytoarchitecture that is a huge challenge to maintain, to support. So axons have very high energetic demands due to constant ion fluxes, synaptic vesicle recycling, transport of cargoes, and maintenance of a large membrane surface area. So what this slide shows is a scaled model of a motor neuron. So a scaled model of an axon of a motor neuron in proportion to the cell body, and here in the distal part, the synapse. And these axons are, so this particular axon is accompanied exactly by 10,000 Schwann cells. So with this picture, I want to show you that this epitomizes that axons are particularly vulnerable and at constant risk of damage. So something can really, because it's so long and fragile, something can really damage constantly in the axon. So therefore, it's not surprising that loss of axon integrity occurs in a broad spectrum of conditions, of neurological conditions. So this ranges from the most classical disorders like Alzheimer's disease, multiple sclerosis, Parkinson's disease, to very widespread current health problems such as diabetic neuropathy and glaucoma. For example, in diabetic neuropathy, many people are not aware of this. 50% of all diabetes patients develop as a complication this kind of metabolic neuropathy. And here I hope you can appreciate that in the late stage of the disease, there's significant axonal reduction due to axonal breakdown. So this also tells you the whole picture that in these diseases, axon degeneration occurs very early, so it's etiologically very important and leads to the most relevant and most debilitating symptoms. Therefore, neuroprotective therapies tailored to preserve axon stability are important for the development of new treatments for these conditions. So some of the best chances, some of the best ways to develop such neuroprotective therapies came from the discovery of components that confer delayed Valerian degeneration following nerve injury. So Valerian degeneration is a simple model to study the mechanisms of axon degeneration, which describes the degeneration or the degenerative events in the distal nerve stem following separation of the axon from the neuronal cell body. So this can be done, for example, in the laboratory mouse by transecting the sciatic nerve. So it's the longest nerve. This is often what we do in the lab. And in a control mouse, in a Y-type mouse, 5 days after injury, so we are looking now here in a distal stump, the axons are completely degenerated, completely degraded. So in contrast, in a spontaneous mutant or also in transgenic mice that express the so-called WLDS protein, or in PHR1 knockout mice or in SARM1 knockout mice, this process is completely disturbed and axons survive much, much longer. So these components, WLDS, PHR1, and SARM1, are intrinsic neuronal components that regulate the inherent axonal autodestruction program after injury. So they confer prominent neuroprotection from inside the neuron. So these mutations, these 3 mutations, they have been introduced in various neurodegenerative models in the last 20 years. But it turned out that neuroprotection could be achieved only to some extent, maybe to a limit, only to a limited extent. only in some of this neurological models, indicating that we need a much better understanding of why axons degenerate in disease, and also we need alternative approaches, maybe non-cell autonomous approaches to protect axons. So as an alternative approach, we ask if we can change the resistance of injured axons to degeneration by manipulations in Schwann cells. So it's completely different thinking. So this question was motivated by earlier studies indicating that Schwann cells provide trophic metabolic support to axons independent from their more traditional myelination functions. So this was published in these papers, including our study, where we showed that exclusively manipulating metabolism in Schwann cells is sufficient to precipitate axonal degeneration. So to test this model, we started off with some very simple experiments. So we used microfluidic cultures in which Schwann cells can be selectively associated with axons, and then the axons can be injured to induce axon degeneration. The speed of axon breakdown can be assessed, can be measured. So here you see the neuron-only condition. So these are the small cell bodies of the neuron. Then the axons, they grow through these small microchannels. So you can separate the axons from the neuronal cell body. So you can induce this Valerian degeneration. And then you can compare this to the condition where the neurons are, or the axons are cultured together with the Schwann cells. So the Schwann cells associate and provide maybe dystrophic support to perturbed axons. So what we found is very intriguing. While in the neuron-only condition, 25 hours after injury, the axons were completely degenerated, we found that the association to Schwann cells conferred significant axon protection, supporting really the notion that indeed Schwann cells stabilize injured axons, likely through their metabolic activities. So what exactly could these metabolic activities be? So to study metabolic changes, changes in intermediate metabolism that occur in Schwann cells upon axon injury, we used our in vivo Valerian degeneration approach, in which the sciatic nerve of the mouse is axotomized. And then metabolic changes can be analyzed in fluorescently labeled Schwann cells in the distal nerve stump that undergoes axon breakdown. So in these nerves, the Schwann cells are green, and this facilitates co-localization studies. So you can clearly see by using immunostaining really what is going on in these Schwann cells after injury. So we screened for a variety of metabolic changes in these Schwann cells, including lipid metabolism, mitochondrial metabolism. But to cut a long story short, what we found is actually a dramatic upregulated expression of glycolytic and fermentation enzymes and their corresponding activities in these distal Schwann cells accompanying axon injury. So these are the processes in every cell that generate pyruvate and lactate from glucose. So you know, glycolysis is a 10-step metabolic process. So there are 10 enzymes involved. And we found dramatic upregulation of literally all these enzymes in injury-activated Schwann cells. Here you see on the right-hand side examples of the first enzyme of glycolysis, hexokinase. And here's the 7th enzyme of glycolysis, phosphoglycerate kinase. So in uninjured conditions, you barely see expression of these enzymes on longitudinal sections. But if we injure the nerve 36 hours after injury, so actually briefly before the axons break down, we see dramatic upregulation of expression of these glycolytic enzymes. So in here you see that there's nice co-localization with Schwann cell bodies. So there's not only upregulation of glycolytic enzymes per se, but we see also strong increases in the expression of central positive glycolytic regulators. So the most important are PFKFB3 and LDHA. So PFKFB3 is known, for example, from the cancer field as a very important glycolytic promoter, and LDHA is actually promotes fermentation, but also glycolysis through the regeneration of NAD plus, which is an electron acceptor. So it generates NADH, so it drives the whole glycolytic process. So here I hope you can appreciate that this is also very early induced after injury in this colocalizes to Schwann cells. So this led us now to propose following model, suggesting that Schwann cells strongly upregulate the production of glycolytic end products, so pyruvate and lactate, through glycolysis upon nerve injury, so before axons degenerate, because they need more energy because they are injured. And these energetic substrates are then shuttled, brought into axons through monocarboxylate transporters expressed in Schwann cells, and are then used and are then metabolized in axons for compensatory energy production to stabilize distressed axons. So we hypothesize that this is a physiological adaptive mechanism, not only in this harsh injury paradigm, if you transect a nerve, but also in other neurodegenerative models. So, and this promotes the bioenergetics of perturbed axons through this pyruvate lactate crosstalk to support the survival of axons. And I show you now on the next slides some preliminary evidence that very much supports this model. And then we did also experiments actually to definitely test this model. So the first thing that is very consistent in this model is when we took Schwann cells in a dish and we injury activated them by activating the ERB2 receptor on these glial cells, which has been previously shown as a very early injury signal. We found that they had strongly increased glycolytic activity and they release dramatically elevated levels of lactate. However, this was a little bit surprising when we looked into injured nerves, but this supports very much our model that this lactate is consumed. We found a totally contrasting result. So the lactate was actually not increased in the nerve, it was decreased. And this indicated to us that the secreted lactate secreted from Schwann cells is very quickly consumed by injured axons. Another observation that we had that support our model was the expression of the monocarboxylate transporters. So the monocarboxylate transporters expressed in Schwann cells are mainly MCT1 and MCT4. So these are isoforms that are known to facilitate the release of pyruvate and lactate, especially MCT4. So they have low affinity for this metabolic substrates in contrast to neuronal monocarboxylate transporters. So very, very similar to this glycolytic upregulation. So I hope you remember this glycolytic enzyme upregulation in nerves. We found also strong increases in the expression of MCT1 and MCT4 in this distal nerve stems, colocalized in Schwann cells, so supporting our model. So this was actually very accentuated for MCT4, which has a very strong activity for release of lactate. We next ask, so if this lactate or pyruvate is really axon protective. So we took DRG cultures, the spinal cord neurons that I also showed before, and we transected the axons under control conditions. The axons degenerate quite quickly. So after 24 hours, as you see here, everything is gone. If we administer a cell-permeable form of pyruvate into the culture, the axons are strongly protected. And this is quantified here. So altogether, so this supports this supports very much this model. But how can we actually mechanistically really test this paradigm? So we pursued two mechanistic avenues. First, we attempted to prevent the strong increase in pyruvate and lactate production and see what happens to axon integrity after injury. So we would predict that this would lead to faster axonal breakdown because they don't get these energetic substrates. And then in a second approach, we also pursued the idea of blocking transfer of this monocarboxylates through inactivation or blockage of MCTs. So we generated a bunch of mutant mice to suppress glycolytic activity and production of lactate in Schwann cells, and then we subvert the nerves. So here you see mice lacking GLUT1 in Schwann cells. So this is GLUT1. Then these mice are missing the positive glycolytic regulator PFKFB3, and these are mice deficient for the glycolytic promoter LDHA. So these are conditional mutants. And it's interesting to know that under physiological conditions, so non-injury conditions, the nerves look pretty much normal. So there's no spontaneous axonal degeneration. However, when we lesion the nerves and we measure the rate of axonal breakdown, we find statistically significant acceleration of axonal degeneration. And this is quantified here. And these results were actually very strongly accentuated in the GLUT1 mutant mice. So GLUT1 is the transporter that brings glucose into the Schwann cells, and the glucose is then used in the glycolytic process to produce pyruvate and lactate. So this very much supports our model of the cell non-autonomous metabolic support of axons. So to test if the suppression of monocarboxylate coupling between Schwann cells and injured axons through MCTs also evokes accelerated axon death, we applied the Schwann cell DRG co-culture exotomy assay that I introduced previously. So when neurons are cultured by themselves and the axons are transected, they degenerate within 24 hours. When axons are cultured together with Schwann cells, they survive clearly longer. So you see more axons here. However, when we apply a pan-MCT inhibitor, so this is a pharmacological approach, so we completely revert the phenotype. The co-cultures, they look like actually neuron-only cultures. So we block actually this metabolic communication, the pyruvate and lactate communication between axons and glia, and we revert the phenotype. So this also strongly supports our mechanistic model. So far, all these avenues that we have pursued, you have seen that this leads actually to faster axonal breakdown. So we tested our hypothesis, we kind of suppressed glycolysis, we suppressed the metabolic communication, and this leads to accelerated axon breakdown. But the next big question for us was, is there maybe something that we can try to boost glycolysis, to increase glycolysis, to enhance this metabolic crosstalk with the final goal to achieve axon protection, and also later with the goal to protect axons in disease. So for this purpose, we carried out a phosphoproteomic screen with the idea to identify, to find signaling cascades that maybe regulate this metabolic, this glycolysis, the metabolic crosstalk between axons and glia. So what we found is very fast activation of the mTOR pathway in Schwann cells. So here you see a Western blot. Phospho-S6 is a widely used readout for mTOR activity in cells. And mTOR activity is already induced 30 minutes after injury. And this really happens in Schwann cells. So this is the same immunofluorescence approach that you've seen before with these glycolytic enzymes. So 36 hours after injury, before the axons degenerate, you see dramatically increased phosphatase 6 staining in Schwann cells, so co-localized in Schwann cells. So why is this mTOR pathway so exciting for us? So the mTOR pathway, as you know, is very well known from the cancer research field to be a key promoter of glycolytic activity. So there's hyperactivation of mTOR in many cancer cells, is kind of hallmark of cancer. And this drives the abnormally increased glycolytic activity and lactate release, which is also known as the Warburg effect. So this mainly occurs through the transcription factors HIF1 alpha and c-MYC, as is shown here. So these transcription factors together control the expression of many glycolytic targets, including glycolytic enzymes and positive glycolytic regulators. So also this PFKFB3 and LDHA. So we generated mutant mice with inactivated mTOR signaling, specifically in Schwann cells. And we first wanted to confirm that really this glycolytic promotion, the glycolytic activity is abrogated in these mutants. So what you see here on this slide is digital droplet PCR. So in control animals and wild-type animals, consistent with this immunostaining results, you see strong upregulation of these glycolytic components in injured nerves. So this is the black line, and this is blue line. And as expected in the mTOR conditional mutants, so mTOR inactivation in Schwann cells, we see that this upregulation is very clearly abolished. So now the central question for us is this consistent with our model? Does the abolished upregulation of glycolysis really results in faster axonal injury after nerve lesion? So, and here on this slide, you see that this is really the case. So we labeled in this mTOR conditional mutants axons genetically in a fluorescent way. So in control animals, 36 hours after injury, you see still many axons that are intact, but in mTOR inactivation mutants, you see that axon degeneration is strongly accelerated, so they are fragmented. 48 hours after injury in electron microscopy, you see the same picture. So the magenta labeled axons are degenerated axons, and the green axons, they are still intact. So this is quantified here also 36 hours after injury. So this is totally consistent with our model. So now the reverse picture. So we were able to accelerate axon degeneration. Can we use the signaling pathway? Can we use the mTOR pathway in Schwann cells in non-cell autonomous manner to confer axon protection? So to test this idea, we exploited a negative regulator of mTOR signaling. So there's a complex which is called TSC1, TSC2. So especially TSC2 is a strongly recognized negative regulator of mTOR signaling. So if we remove it from Schwann cells, we induce mTOR hyperactivity. So here, this you can appreciate in this Western blot. So we remove TSC2 in Schwann cells, and we see the sustained mTOR hyperactivity in Schwann cells. So this is measured here. And now the big question, does this result in axon protection? So we injured the nerves of these conditional mutants, and totally, as anticipated from our model, for the first time in a non-cell autonomous manner, we see that this can confer axon protection. So this is 48 hours after injury. In control animals, the majority of axons are broken, and in the TSC2 knockouts, there's significantly increased axon survival. So this is quantified here. So to test this, if mTOR hyperactivity can also confer axon protection in a more subacute form of axon degeneration. So what we have seen previously is acute form, very harsh injury. This is now subacute. So we applied a neurotoxin-induced neuropathy model. So we treated mice with the neurotoxin acrylamide, which is known to induce axon degeneration and several neurological abnormalities already after 2 weeks of treatment. So what you do here is you put acrylamide in the drinking water of mice. So here you see this acrylamide induces in a more asynchronous pattern, in a more mild pattern, the activation of mTOR signaling. So showing that this is this adaptive response of Schwann cells. So they upregulate glycolysis through activation of mTOR signaling. So we wanted to see if we can confer neuroprotection, axon protection in this model through preemptive overactivation of mTOR signaling in Schwann cells. So we again exploited our TSC2 model. So as you see here, they have this increase mTOR signaling in Schwann cells. So there's a huge difference between these two pictures. And indeed, we found that the number of degenerated axon profiles in the TSC2 mutants is significantly reduced compared to control animals treated with acrylamide. So we can really achieve neuroprotection in the subacute model of neurodegeneration. So intriguingly, this does not only translate into structural axon protection, but also in improved behavior of the mice. So I hope you can appreciate here. So this is this treadmill analysis, rotarod analysis. If you give acrylamide to mice, they get quite quickly sick, and the rotarod performance, the treadmill performance decreases rapidly. And this is significantly ameliorated in the TSC2 mutants with increased mTOR signaling in Schwann cells. Here you see grip strength analysis, which also strongly alleviates the symptoms. So this is all statistically significant. So this is now unpublished data. So we finally set out to study if the manipulation of metabolic axoglia coupling impacts an established genetic model for myelin-related axonopathy. So this is more a form of chronic axon degeneration. So what we've seen before is acute and subacute. Now it's more chronic axon degeneration. So we used so-called P0 knockout mice. So these animals are known to develop dysmyelination during development, but also demyelination later in adulthood. And all this leads to lower axon numbers in nerve sections. So we quantified this in plantar nerves. So already at 4 weeks of age, we see dramatically reduced axon numbers. So there's very early axon degeneration. And this gets slightly worse with increasing age. So in this model, we could really not test all of our hypotheses. So we cannot really confer axon protection for something that is already gone, but we can actually ablate mTOR signaling to see if actually the axonopathy becomes worse. So what I want also to say here, we were not able to induce mTOR hyperactivity very early, so preceding this 4 weeks of age, because mTOR activity increase during development itself induces changing in the nerve environment. So we were only able to test what happens if we actually inactivate this axoglial metabolic coupling. So here you see that this axoglial metabolic coupling is really active in this disease model. So these are control animals. This is the phosphor-6 staining. And in the P0 knockout animals, you see strongly upregulated phosphor-6 staining in longitudinal sections of the nerves. So the axoglial metabolic coupling, so this increase in glycolytic activity, is really upregulated. So we can ask now what happens if we inactivate this axoglial metabolic coupling. And this I show you on the next slide. So these are videos. I hope that you will be able to see the videos. These are control animals, so the simple P0 knockouts. This is at 3 months of age. So they have behavioral abnormalities. They have a kind of funny gait. They have gait deficits. I try to maybe switch on the movie now. So if you pick them up, you'll see this kind of gait abnormalities. But then if you look at the P0 knockouts, in which specifically, so exclusively in Schwann cells, we inactivate the mTOR signaling, so this axoglial metabolic coupling, then the phenotype, the gait abnormalities get worse, and the animals have actually difficulties even to move. So how does this correspond now to the structural phenotype? So we quantified axon numbers in plantar cross sections, so similar to what I shown you before. So consistent with this picture here, the number of surviving axons in the mutants with deficient axoglial metabolic coupling were significantly reduced. So to wrap up, so all these data together led us to propose the following model for this metabolic support mechanism of perturbed axons. So when axons are stressed, so not only transacted, but there can be also this kind of micro lesions in neurodegenerative diseases. The Schwann cells, so this is the Schwann cell compartment, the Schwann cells are able to rapidly sense the axon injury. Another project in my lab actually tries to define how Schwann cells sense this kind of axon injury very rapidly. So they are rapidly sensing the axonal perturbation. They upregulate the glycolytic activity through the mTOR pathway. The Schwann cells produce increasing amounts of pyruvate and lactate from glucose. These substrates are then transported through the monocarboxylate transporters 1 and 4 into the axonal compartment. So the axonal compartment has actually predominantly the MCT isoform expressed. The lactate then in axons is reconverted into pyruvate through LDHB. And then the pyruvate is used in axonal mitochondria to generate energy to counteract the axonal deterioration and the axonal breakdown. So this is the reason why the axons survive longer. And can actually also recover from injury. So this kind of alternative form of axonal regeneration that I touched upon at the beginning of my presentation. Okay, so that's it. This is my acknowledgement slide. So I want to especially acknowledge the main driver of this project in my lab, Elisabetta Babetto. So Dr. Babetto, she basically spearheaded the project. And I also would like to say a big thank to our funding source, because these are very expensive experiments with all these transgenic animals that have to be kept in the animal house. Thank you so much for your attention, and I'm happy to take any questions.
Okay, perfect. So someone wants to ask? Maybe I can start.
Javier had, I think, a question.
Francisco?
There's friend. Okay.
Yes.
I have a question. Yes. Thank you very much for this talk. It's really very good. I really enjoy it. I have a couple of questions concerning the. I mean, like something like a little bit more in the theoretically on the concept. Is possible also that in the remarkable bundles, so where you have non-myelinating Schwann cells, such as mechanisms also cure? I mean, you don't need probably proper myelination there to have the same interaction between Schwann cells and axons. This is first question.
Yes.
And second question is something I will ask you later. Let me know.
Yeah, the question about remark bundles, so unmyelinated axon, it's very interesting. We did not look specifically into remark bundles, but what I can tell you from earlier studies that we have performed, so in Valerian degeneration, somehow the small diameter axons, they degenerate faster than the larger axons that are myelinated. So I showed you that that in Valerian degeneration, the axon breakdown starts something like at about 40 hours. So I think this is more true for this larger diameter axons, so myelinated axons. As you know, because the small diameter axons, they are much smaller, they're more difficult to investigate. You would have to do more electron microscopy studies. And also in these nerve sections, you know, if you use the genetic labeling, it's not clear if this represents unmyelinated or myelinated axons. So I can't really tell you for sure, but we think that the mechanism outlined here is also true, is also relevant for the protection of remark axons. But we don't have really super good evidence for this.
So just that I saw some of the axons there in your electron microscope pictures. I will see some of the small axons there, so I thought, okay, maybe this is something there. And the second question I have is concerning, you know, this kind of possibility that mitochondria can be transferred as well. So if
from Schwann cells into axons.
Exactly.
Yeah, the work from Philippe Court and so on, and from Jan van Minnen, and So I mean, we never looked into this transfer of mitochondria. My understanding is from, you know, listening to these talks from other people and from talking to these people that discovered the mechanism, is my feeling is that this is a kind of sporadic mechanism. So the amount of transfer is, I don't want to say it's minimal, but it rather occurs, you know, through very long periods of time. And what we are looking now in my studies are the very rapid changes. So, I mean, we did not look if the transfer of mitochondria is increased after injury. So we have no evidence really for this. We have strong evidence for this increase in glycolytic enzymes, and actually we looked also in mitochondrial metabolism. We don't see, you know, increased activity of Krebs cycle enzymes. So so this is totally in contrast to the glycolytic enzymes.
Yeah, that's why I was asking. But thank you. Thank you very much. Very interesting.
Okay, Javier.
Yes, thank you, Bogdan. Super nice talk.
Thank you.
Super, you know, round results, like, you know, the complete whole story. My questions are from a person that is not in the field, in the Schwann cell field. So I have two questions. One is, which are the signals from the axon to the Schwann cell to, you know, change their metabolism in the way that you've shown? This is the first question. And the second question is, which are the things that are going on in the Schwann cell besides the ones that you show? I mean, are the Schwann cells reacting to the damage in the axon? Are they expressing, I don't know, inflammatory factors or signals for the rest of, you know, the innate immune system?
Yeah, these are very good questions, central question for our lab, especially the first point that you mentioned. So how do Schwann cells sense injury? How is the injury signal, you know, induced in this glia? I little bit alluded already to this. So I mentioned this activation of ERB2, ERB3 heteroreceptor pair. So this receptor, you know, is known since 20 years, and it controls all different biological aspects in Schwann cells. People know that, you know, this regulates Schwann cell proliferation, myelination, survival, basically everything which is important for the development of nerves. But we found it very intriguing, and our paper puts, I mean, is actually the paper Nature Neuroscience is based on this observation. We think that there's very early activation of ERB2 of this ERB2 receptor. We find it actually 30 minutes after injury activated. But there's another paper in Journal Neuroscience. They claim that it's already activated like 5 minutes or 10 minutes after injury, and they show that this is takes really place in Schwann cells. We don't really know, but this is one project that tries to exploit to explore this. We don't know which factor activates ERB2 on the surface of Schwann cells, but ERB2 is known to drive PI3 kinase signaling and also MAP kinase signaling. And MAP kinase signaling and PI3 kinase signaling, together with downstream AKT, they are known to converge of mTOR pathway. So we think that these two signaling arms leads to activation of mTOR in Schwann cells. So I hope this answers a little bit your question. So maybe for this injury signaling, I mean, it is known that ERB2 is activated by neuregulin. So there must be something in the axonal membrane, maybe fragments of the membrane after injury that are released. They go to this receptor. But the big question for us is, how is it possible that Schwann cells, centimeters away even from the injury site, can sense this very quickly? So it's not really a simple explanation for me how neuregulin could do this. Maybe it's there's also a kind of electrophysiological explanation behind this phenomenon. So at the moment, this is a black box for us.
I'm sorry, and the other part was, which are the changes in Schwann cell besides the changes in metabolic support?
You are absolutely correct. So, I described this Valerian degeneration process. So this is a more holistic term. Valerian degeneration is not only the breakdown of axons. I did this a little bit simplified for my presentation, but Valerian degeneration encompasses also the reaction, the behavior of other cell types, of course, of Schwann cells, and later both resident macrophages and invading macrophages. So what we looked in our study are very early changes in this interaction. So 24 hours, 36 hours after injury. In our experiments, we don't see really activation of macrophages at so early stages. They more start, let's say, at 72 hours after injury. Therefore, we think this question is not so relevant. So in our time points, we don't see actually many macrophages. They come a little bit late. So, but I agree, for later, effects of Valerian degeneration, macrophages are very important to clear up all this debris, then C-Jun signaling in Schwann cells and facilitation of regeneration. But I hope that you could maybe recognize in my presentation that we believe that there are two distinct neuroprotective mechanisms in Schwann cells. First, our mechanism to prevent axon death, and later, this neuroprotection by growing of new axons through orchestrating C-Jun.
Yeah, okay, thank you.
Yeah.
Patricia?
Thank you very much, Bogdan, for such an interesting and amazing talk.
Thank you.
You convince us that the glycolytic activity is essential for the neuroprotection of the axon. But I have some questions about what happened with the phenotype of these Schwann cells when you modify and you hyperactivate the mTOR signaling.
Yeah. So there was this a related question. So not only the mTOR signal, but what happens if we inactivate this glucose transporters and PFKFB3 and LDHA? A very interesting question. So regarding the glycolytic components, as I mentioned briefly, we don't see much change except in GLUT1 animals. So the GLUT1 animals, they have slightly thinner myelin. So it may indicate that this kind of glucose metabolism is also somehow implicated in myelination. But the phenotype is not very prominent. If you look at the nerves, they look very, very similar, like wild type nerves. It's only if you measure very accurately the G ratios. And actually, we publish this in this paper. With mTOR animals, I'm sorry that I did not explain this very well, but I think it's an important point. The Raptor mutants, so we use the isox10-CRE driver. So you have to treat these animals with tamoxifen to induce the recombination. They don't have any phenotype. They show totally normal nerves, totally normal myelination. So this is relevant for the studies that I showed here. So without any other intervention, there's absolutely no change. Nerves look normal. However, if you stress the axons, then it gets worse. So it precipitates more axon degeneration. So yeah, I think this is the answer.
Okay, Patricia, something else?
Okay, sorry. I thought he was trying to put again the video. No, thank you.
No,
it's okay.
So I just wanted to say that under normal condition, just with the mTOR inactivation, you don't have any perturbation of the nerves. They look normal. So I think it's an important point to mention, because some people might say, yeah, you make the phenotype worse because you destabilize the nerves, but this is not the case. The nerves look totally normal. Yeah? You only change it in injury conditions.
Mm-hmm. But when you show the results in Valerian Degeneration, I didn't see so much proliferation of Schwann cells like in other models of Valerian Degeneration. That's why I'm thinking perhaps it's not the only pathway involved in the regeneration, because I don't see here, not so much when you show the expression of the glycolytic enzymes, you show the DAPI nuclei, and they are not so proliferated as in a normal Schwann cell Valerian Degeneration model. And also, you don't show so many changes in the axons when you speak about the glycolytic activity. So my question is, if there is, perhaps this is one of the items involved in the protective mechanism in this way. For example, when you see that the glycolytic activity may be one of the mechanisms involved in the protective action of the Schwann cell on the axon. But what happened in a model that it's more severe, when you see a much more degeneration? It's only, it's enough to recover, or you need another mechanism also that participate together with the metabolic activity support for the regeneration?
Yeah, so I think I have to address the first question, because I think there is a little bit of misunderstanding. So in my lab and also in all these papers, we don't study axonal regeneration. So to explain, because most people are not aware of this, we are studying axon degeneration. So all the processes that precede, axonal regeneration. So here you have the neuron, here's the cell body. So then you transect an axon, and the distal part degenerates. It doesn't regenerate. So the distal part degenerates, breaks down, or dies. And we are looking in my projects what can prevent or what influences the axon stability, what influences the degeneration of the axon. So what you are talking about is the proximal axon stump that actually after injury regenerates. So we are looking only in the distal stump. So this is often confused.
I am asking, no, sorry, perhaps I didn't explain myself. But what I speak is what happened then. If you consider that in the distal stump, if you have a model of crushed sciatic nerve where you can regenerate the axon, what happened after this period? If this upregulation of the glycolytic activity may be enough to promote regeneration.
Yeah, so I mean, if the axon is repaired from like smaller lesions, you know, the glycolytic upregulation is only transiently observed, then it goes down, nothing happens. I don't think that nothing needs to happen because the axon then is fine. It recuperated from the injury and nothing needs to regenerate. So this is my answer to your question. So a completely different question, I think maybe this is what I understand from you, is if the distal part cannot recuperate, degenerates, it's completely gone, and then we need to regrow a new axon. Then one could ask, is the, you know, the metabolic support also required, important for the growth of a new axon? So this is another interesting question.
Mm-hmm.
Yeah.
Well, thank you very much.
Yeah.
Okay, so then there is a question about this mitochondria interchange that I think it was already answered. I don't know, maybe there is any aspect, iPad 2 that wants to address or ask. Anyway, we can go further. And then Brian also has a question, but has a problem with the microphone.
Mm-hmm.
So maybe he can write it in the chat, or maybe it's working now.
So there's a chat function in
Yes.
Zoom.
Yes.
So yeah, but I don't see any highlighted. I don't see any chat at the moment. But I mean, if you can read it.
I will read one question that has Mauricio. I don't know, Mauricio, if you can ask yourself, it's better, but I can read the question also. I can read it. So I have a question regarding the model of hyperactivation of mTOR. Mm-hmm. Did you check the autophagy activation after nerve injury in this mutant?
Yeah, this is very interesting. So this is actually a paper that we are working currently on, because autophagy is also heavily involved in metabolism and recycling of energetic substrates. So I think Chris and Jason's group, they have shown that you have very early and strong upregulation of autophagy. This is actually a little bit surprising if you think about the mechanism behind this. So mTOR signaling and autophagy are antagonistic. So I hope you know what this means. Antagonistic means that mTOR actually suppresses autophagy, so you cannot see at the same time upregulated autophagy as well as mTOR signaling. But surprisingly, we see, you know, exactly the opposite thing. Both mTOR signaling and autophagy is super strongly induced after injury. So there's a little bit of a conundrum now how to explain this, but we are looking in our current project if autophagy manipulation in Schwann cells can also influence the rate of axonal degeneration. So I'm not studying axonal regeneration. Again, my focus in our project is axonal degeneration. Most people, they have a little bit of problem understanding this.
Okay.
So this is what we are currently looking at. Yeah.
I have a couple of questions. Sorry?
My question was if he has checked on later time point, farther than 48 hours.
About autophagy?
You can see a difference, yes.
I mean, yes, at 48 hours, there's strong autophagy induction in Schwann cells, but I can't tell you now what the implication for axon integrity is. So, yeah. You know, Kristen Jessen, they have looked what is the implication for axonal regeneration, and they showed that regeneration is delayed. But we are looking here at a completely different thing. We are not looking at regeneration. We are looking at earlier things. We are looking at degeneration. So
you didn't see the death of the myelinating Schwann cell as well.
Death of myelinated Schwann cell? No,
well, they eat their own myelin, so they activate the autophagy too.
Yeah, they activate the autophagy, but I don't think it has anything to do with death. They activate the autophagy to digest the myelin remnants, to prepare a terrain that is good for axonal regeneration. But again, we are not studying axonal regeneration. We are studying degeneration. Yeah. Okay, great.
I have a couple of questions, Bogdan, if you have time.
Yes, sure,
Yeah,
sure.
okay,
Go
perfect. Yeah, so one is if glycology in one cells is related to this dedifferentiation of the cells.
Yeah, this is actually, this goes hand in hand with the autophagy. So c-Jun and autophagy, they are, implicated in this repair phenotype of Schwann cells, dedifferentiation. So this is one neuroprotective function. It's important for growing of new axons. But as I said, again, we are not studying this. We are studying things that come earlier. So an interesting question is the dedifferentiation pathway, does it have anything to do with the neuroprotective role that is our focus? So we are doing now lesions in c-Jun animals, in autophagy animals, and some other animals, and we want to see if there is changed speed of axon degeneration. So this is what we are currently looking at, and I hope that you will see the paper hopefully beginning of next year. Yeah.
Perfect. So then in the pictures you showed, clearly the Schwann cells are the cells are expressing these glycolytic enzymes, but there are most of the cells, but there is few cells that seems not to be Schwann cells.
Yes, yeah.
So
So
which kind of sensor?
Maybe it's a kind of asynchronous response, so we don't see the upregulation in every cell. So maybe I can go back to these pictures. Maybe here you're right. Here's so the Schwann cells are labeled by PRP, GFP. Here's one that does not show this. So, so what I want to say is, first of all, axonal death is not synchronous in the nerve. Not all axon in the nerve, the whole population, die at the same time. There's only a subset that degenerates, others are still intact. And only the Schwann cells that are touching degenerating axons, they upregulate the glycolytic activity. So this could be an explanation. Okay, then another explanation could be, later the Schwann cells, they cease the upregulation, they downregulated. So maybe the Schwann cells here, they are associated with an axon that is already dead. So this could be an alternative explanation. Yeah.
Then the last question, maybe, in these experiments that you showed that the axon survival is increased with some of these experiments, have you tried after 48 hours? Because it can be that it is kind of delayed, the degeneration. It's not that it is abrogated.
Exactly, it's not completely blocked. In all these experiments, all axons will eventually die. So there's just a delay. So as I said, the Valerian degeneration model is a very harsh model. There's no, you cannot keep axons for, 2 months alive. Everything will eventually degenerate. We are talking here about the rate, the temporal pattern. So so I hope this answers this question.
Okay, Bogdan, I think we are done.
Great.
So thanks a lot. It was a wonderful talk. Very interesting. Something that I didn't know about. So it's very nice, very nice.
Thank you very much for attending and for all these fascinating questions. And I hope we can keep in touch. Yeah.
Yeah, of course.
Thank you very much.
Yeah. Have a good day. Bye-bye.
Have a good day. You too. Bye-bye.
Bye-bye.
Bye.